Okay, so after we have pushed substances through the filter in the glomerulus, we now have a bunch of substances in the filtrate. Now, this fluid filtrate is going to be moving through the proximal convoluted tubule, through the loop of Henle, and then finally through the distal convoluted tubule before finding a collecting tubule and a collecting duct. Now, associated with the proximal and distal convoluted tubules, loop of Henle, and the collecting duct, we have all of these capillaries that are going to contain the blood that left the glomerulus, which means they're going to be running kind of associated with these. So that means we can actually pull substances from the filtrate back into the blood should we need them. Now, this is going to be really important for us for something like glucose or sodium, potassium. These substances, we've spent a lot of time and energy in making sure that we got them to the blood. So we actually don't want to lose all of those. Instead, we want to reabsorb as many of them as we can. Now, some of these substances may also be something that we want to get rid of entirely, something like your ureas, so things like metabolic waste products. We want to get rid of that stuff as much as we possibly can. So we want to concentrate that. But at the same time that we're concentrating something like urea, we actually may want to recover a fair bit of water. We may want to recover all of our glucose. We may want to recover some sodium. So we have all of these things that we can actually regulate how much we are getting rid of and how much we are pulling back in. Now, we're going to be regulating that by figuring out how much we are going to reabsorb or secrete, either through reabsorbing in the tubules or secreting back into the tubules. And we're going to be doing this, like I said, in the proximal, distal, convoluted tubules and in the loop of Henle. Now, one of the reasons that we can do this is that we have running alongside our proximal convoluted tubule, we do actually have these paratubular capillaries, which are going to be fairly close. Okay. Now, tubular reabsorption is going to be when we're moving substances from the tubule into the blood. Tubular secretion is when we're going to be taking substances from the blood and putting it back into the tubule. Now, oftentimes this is going to require some sort of transport. Sometimes it will rely just on open pores. So whether it's like an aquaporin, which will allow water to move freely, and just kind of relying on diffusion. Other times we might need active transport processes. So if we're trying to really concentrate something, we may need to be using some active transport just to make sure that we can move substances against a gradient if we do need to. We're also going to be utilizing some pinocytosis or exocytosis of some of these molecules as well. So we're going to have a lot of variance. And that variance is really just going to depend on what it is we are trying to move. Now, the first one that we're going to look at is glucose. The reason we're going to look at glucose first is because glucose is special and that we actually want to reabsorb all of it. Like I said earlier, we've put a bunch of effort into getting glucose into the blood. And all of our cells need it. So we don't want to waste that precious resource. So instead, we want to reabsorb as much of it as we possibly can, preferably all of it. And in particular, we're going to absorb all of it in our proximal convoluted tubule. However, you would imagine, though, that we're going then from an area of somewhat low concentration of glucose into an area of high glucose, which means we have to have some sort of active transport to do this. That means we're going to have some proteins in the microvilli of the proximal convoluted tubule that is going to allow this to occur. Now, this is also a great place for you to realize why we need those microvilli. That's because we're relying in many cases on transport proteins or proteins embedded in the membranes of these to allow us to move substances back and forth, which means we need more room to have more proteins. If we need more room to have more proteins, we have to have more surface area. And that's why we have these cells are going to be covered in lots and lots and lots of microvilli. But this way, by using this active transport, we can actually get this number down to roughly 0 under ordinary circumstances in a healthy individual. Now, I say ordinary circumstances because we know that there are maybe times when there is massive amounts of sugar that would be found in the blood. So say you've eaten a gallon of ice cream or something like that. So you have massive amounts of sugar. If the concentration of sugar in the blood is over around 375 milligrams per microliter, I think I have the volumes wrong. If the numbers are too high, we may not be able to reabsorb all of it. However, we're still going to do the best we can. So someone that has something like, or a disease like, say, diabetes, where they're going to have really, really high concentrations of glucose in the blood, well, those folks may not be able to reabsorb all of that glucose just simply because there will be too much of it found in the blood and thus found in the urine filtrate. However, this also means if we can't reabsorb it all, that in the end, you would actually find glucose in that person's urine, which is actually not something that we expect to see in a normal, healthy individual. So this is oftentimes why finding glucose in the urine of a patient is actually indicative of a disease like diabetes. Now, we're also going to be reabsorbing as much of the protein as we can. Now, we shouldn't find very much protein in the tubular fluid at all to begin with because of all of the layers of filtering. However, sometimes things do slip through. So some small proteins and even some moderate sized proteins may actually be able to accidentally go through the filter. Now, proteins are fairly large, though. These are not little tiny single atoms or handful of atoms of things. Instead, they're going to be big. So the way we're going to take those in is we're going to bring those in by endocytosis, and then they get broken down. They're going to get broken down by lysosomes, which contains enzymes, the lysozyme enzyme, which is going to break down those proteins into amino acids, and that's the amino acids that get pulled back into the blood. Now, there's also going to be lots of variance here. That's because some stuff we want to regulate. The glucose reabsorption isn't even something we really tend to regulate. It's just going to happen. We're just trying to absorb as much of that as we possibly can, regulation or no. However, that's not the case for the vast majority of things. That's because our body, remember, is incredibly well regulated. Okay. So something like sodium might be important for us to regulate. Like we do want to reabsorb a bunch of it. However, we may not want to reabsorb everything. So say you had a massive meal at Roadhouse and you had tons and tons of salt in your blood. You don't need all of it back. So you don't mind getting rid of some of that excess salt in the urine. So we're able to do this by regulating how much of the sodium gets reabsorbed in the collecting ducts and collecting tubules over here, whereas most of it gets reabsorbed in the proximal convoluted tubule, a little bit in the nephron loop, so around 25% of it, and then a little bit more in the distal convoluted tubule. We can then start to regulate within these parameters, like, okay, how much are we actually going to need to reabsorb? And that's going to be based off of body's need and how much is in there. The same thing is going to be true for water. If you're drinking a bunch of water and you're staying well hydrated, you don't need to keep all of that water on board. So instead, we may need to regulate how much of the water we are going to reabsorb from that filtrate. Most of the water we will reabsorb, and this is because we need it, and we need to make sure that we get blood volumes back up to where we want them to be. However, we don't necessarily need to reabsorb all of it. So this is, once again, give us a great place to try and regulate this. Now, what we're going to be using to regulate this is actually going to be antidiuretic hormone. Antidiuretic hormone, antidiuresis means not to get rid of water. So we're going to hold on to water in this case. So we're going to hold on to water by basically making more aquaporins. More ADH means we're going to secrete more aquaporins so that we can take in additional water back into the blood, meaning we're going to have more concentrated urine. Potassium is something else that we might want to regulate. Potassium is going to be very important in making sure that nerves and muscles and everything like that function correctly. However, we also don't need too much. Once again, we're going to absorb the vast majority of it in the proximal convoluted tubule and some in the loop of Henle. But then once again, we're going to start regulating this, and we're particularly going to be regulating over here in the collecting tubules and collecting ducts. Now, different hormones can be used to actually regulate how much of these materials we are reabsorbing and the places that we are reabsorbing them. Okay, so a good example of this is parathyroid hormone. Parathyroid hormone, when it's secreted, it's going to inhibit how much phosphate we're going to reabsorb. Okay, so we're going to reabsorb less of our phosphate in our proximal convoluted tubule, and it's also going to stimulate reabsorption of calcium here. So, well, if we're getting rid of a bunch of negative charge and we're bringing in a bunch of positive charge, you might see that we're actually going to be using this to help us regulate pH. We can also regulate pH, of course, using bicarbonate ion and hydrogen ions. So most of this is going to be happening in the proximal convoluted tubule, but then we can start to regulate everything over here in the collecting ducts and the collecting tubules. So this is also how we might regulate, say, the pH of our urine, while also kind of regulating the pH of our blood.